Okay, and while difference to normality, I'm recording on the cell phone first, and now I'm going to start recording on the other thing. 3, 2, 1, now. Well, where was I? Where was I? I didn't even throw in my title today. Gosh. Let's get back into it. So, first, live update my note like a professional. Now you're getting the real Mr. Kind of Marsh experience. That is for sure guaranteed, 100%. Okay, title fixed. Introduction to biology. Summer 24. This is our fourth section. Behavior and genetics. Behavior and genetics. What more can be said? These are pretty much the coolest things going on. Behavior and genetics. This is a mouse. This is a mouse. I've given this mouse a name in the past. It has many names. You may choose a good name for it if you would like, but its scientific name, or rather, first, its common name would be Oldfield. It's an Oldfield mouse. And its scientific name would be Peromyscus polionotus. That is, of course, Peromyscus polionotus. We've known this guy as Peepaw. Peepaw. Peepaw, or the Oldfield mouse. Very cool. It lives on a beach. You'll like it. You'll see it digs actually quite distinctive, long entry tunnels with long escape routes. And that's actually going to become important to us in just a minute. I bang my thing all right. Lives on a beach somewhere. This with a spot on it for some reason is also a mouse. This is a deer mouse. And it also has a ridiculous scientific name, Peromyscus maniculatus. Though, of course, we know it as P-Man. P-Man and Peepaw. P-Man lives everywhere. Including most of the US and Canada. The Oldfield mouse digs a tunnel like this. The Oldfield mouse digs a tunnel like this. Excuse me. Let's toss a ladle in that just in case. Whereas the deer mouse digs a tunnel like this. Notice we have an exit or an escape route We actually, though I've done a pretty poor job of depicting it, I'll fix it there. And we have quite a long entrance. And here we have quite a short entrance. And noticeably, no escape route. So these are two different behavioral differences between these mice that seem to be very consistent. Right? We observe them consistently. Once we find a particular type of mouse, we even are so sure about these sorts of things that we often will claim a particular animal is present solely based on the type of burrow that matches it, right? You know, and a range, of course. So once you're dealing with these factors, you know, you're dealing with really deterministic sorts of stuff. This is a sort of mouse. This is how these kinds of mice act, and they're very consistent in nature, it seems, unless there's some severe environmental pressure or threat. So this is interesting that these mice perform this in different ways. Part of the reason that I've included their habitat information is there's some explanation in that here. These beach-living mice are obviously in habitats that have tons of other fresh food and fresh life available to predators. So these are things like seagulls, which will snap up absolutely anything that they can get at. And so having an exit route, having a deeper burrow is obviously really important for these mice. Whereas for these mice that live pretty much anywhere, just the flexibility and the speed of which you can create a burrow in what for you are more likely to be changing circumstances, especially as a species, is perhaps a more important genetic factor. It's really fascinating there. So, if you put a deer mouse with an Oldfield mouse, they'll love each other very much. And they'll have kids. In biology, we call this crosses, as discussed before. And we call their kids crosses of the parents. This first cross would be, does anybody know? Can anybody remember? An F1 hybrid. What we didn't talk about last time we talked about F1 hybrids was if we take our F1 hybrids and we cross them with more F1 hybrids, what we get is F2 hybrids. And if we take our F2 hybrids and we cross them with F2 hybrids, well, by golly gosh, you start to see a pattern here, right? We get F3 hybrids. If you take our F3 hybrids and cross them with our F3 hybrids, why, the result of that could only be F4 hybrids, which is all totally crazy. So yeah, we rarely have to deal with that sort of thing, but it's important to understand how basic that relationship is and how it works. Right? That's how your naming works. Once you start crossing F1 hybrids and F2 hybrids, you're dealing with a very different thing. You're no longer dealing with F3 hybrids. So, what color do I want? The reason we call some crosses F1 hybrids is because they come from distinct genetic lines. F2 hybrids are then the crosses of two distinct genetic lines, etc., etc. Now that we've taken a quick refresher look at the genetics, let's take a quick look at the behavior. The F1 hybrid excuse me. The F1 hybrid had tunnels that looked like the Oldfield Mouse tunnels. So if you remember properly, those are the ones with escape routes and with long entrances. Long entrance and escape. So that's interesting. The F1 hybrids of the Oldfield Mouse and the deer mouse, those guys have long tunnels. But then, we crossed the F1 hybrids back again with the deer mouse. F1 hybrid cross deer mouse. And this back cross produced 4 distinct behaviors. These are the 4 distinct behaviors that we looked at the genetics for last time, where we're sort of producing these distinct changes in the genetic code and the chromosomes of these different sorts of species. All of that maps directly onto what we're doing with these mice right now. But as opposed to going through it from the perspective of the actual chromosomes, this time we're looking directly at the behaviors. So this backcross produced 4 distinctive behaviors. I'll redraw the mouse. Could either do one of these 4 things. Could either do the complete deer mouse behavior, long exit, long tunnel. Could either do a short entry tunnel with an exit, so an interesting mashup between the 2 behaviors, or it could do a long entry tunnel but then no exit, or it could do just a standard Oldfield Mouse tunnel. So this is basically the complete combination. Here we have the exit tunnels, the escape tunnels. Here we have no escape. Here we have short entrances. And here we have long ones. And you'll see if you make a little cross of traits like this, this axis is short, this axis is long, this axis is escape, tunnel, right, and this is no escape. So we have all the different possibilities covered here, which is really cool. And what's even more interesting is that those come across according to a proportion that you can estimate based on genetics. So these children are really special. and they make for a really cool implication. If we can read a creature's code, its genetic code, we might be able to predict its behavior. So this creates a real motivation to understand chemistry. You have to understand some basic chemical principles in order to understand the biochemistry required in order to understand genetics. Otherwise, other people might be able to get like some sort of sample of your flesh and understand what you're going to do using some sort of computer or just by writing it out on a piece of paper, and you won't even know. Or at least that's the possibility that exists. We'll take a deeper dive into the actual chemistry required to understand this stuff, and of course continue looking at all the fascinating different sorts of biology that ultimately unravel whether or not that's possible, and definitely whether or not it's a good idea. Catch you next time, and keep on learning.